So I don't know how much you know about me, but yeah, I'm running a little YouTube site here, and Answers in Atheism. And my name is Mike, and or Speed of Sound, so you can call me Speed or Mike. And I advertise myself as a high school dropout, because I did in fact drop out of high school, even though I ended up doing a little bit of college after that. But I came up in programming and electronics, digital electronics, but I've always had a passion for and a background in organic chemistry, molecular biology, and the sciences. So I kept that on the back burner until around 2001, 2002. And I got fairly serious about self-education. So most of what I do is trying to self-educate myself in molecular cell biology. And, I think I've done pretty good. But of course, when you self-educate, you never know where you're at. So anyway, but we run this atheism site that's primarily directed at pseudoscience in young Earth creationists. I don't know. Are you familiar with them?
No.
Okay. Okay. Young Earth creationism and fundamentalism is kind of a big deal in the United States. And there's a pile of them that are putting out a whole lot of misinformation about science, and they deny all of geology, half of physics, all of radiometric dating, all of evolution, and all of biology. So anyway, we end up in fights with those guys quite often. And that was in fact, how I found you. One of them was talking about your paper, and he was talking about how many petabytes the brain stored, and then he said, and how code-like it was. And his whole idea is that the brain is a computer, or the brain in the cell is code and computer, and it was designed primarily by God, just exactly like man would design a computer program and would design, that sort of thing. So he had your paper on the screen, and I thought, oh my goodness, a new memory system, right? And I started listening to it, and of course, my first reaction was the same reaction I had to Roger Penrose and Stuart Hameroff. I don't know if you're familiar with their microtubules and quantum mechanics. My reaction was, I'm pretty skeptical, right? But then the further I looked into it and having a short conversation with you, I became, I saw something, and and so I guess I would like to have a conversation about what I saw and what you think it is. So anyway, I'll let you introduce yourself.
Okay, yeah. So my name's Dr. Ben Goult. I'm a reader, which is the equivalent of a associate professor, based in the UK at the University of Kent, which is in Canterbury. And I research into a field called mechanobiology, trying to understand how cells can sense physical parameters, sense the environment, and use that to determine how they respond and how they behave.
Okay, you've been doing this, Talin. Am I pronouncing that right? Talin research for quite a while.
Yes. So okay. I moved into, I started working on Talin in 2006, where I was hired by a guy called Dave Critchley at Leicester in the UK. And he was the big Talin guy at the time. And when we came to work on Talin, it was just seen as this essential adapter for how all our cells attach in the right place in the body,
right?
Helping to hold us in place. So when we started back then, we had no idea where we've ended up more recently with the MeshCode theory, which I'll talk about probably over the next little bit of time. But we're very interested. This protein is really big. It's really conserved, like every single animal from slime mold and beyond, even all the way up to us. We've all got the same version of Talin, multiple Talins in some cases, but the same switch pattern, the same arrangement. So for some reason, nature has kept this, values this molecule highly because it's kept it so well conserved through time.
Yeah, and that's probably the first thing that really struck me is the amount of conservation. I mean, that is, there's something up there. And
but there's lots of these big adapter proteins, but over time they usually change their length. Some get a bit shorter, a bit longer, because if it's just an adapter holding two ends, it doesn't really matter if it's that long, or that long. But here, each of the switches are in exactly the same order all the time. And throughout, even in slime molds, you can see the same 13 switches. So it's highly, highly conserved at all levels, from the amino acid level to the protein domain level to the function.
So at the at the level of just, you know, any tissue in your body, basically, how are those switches? How is this what is the possibilities of mechanical memory, like for two cells, you know, basically smushed together? What you know, with pressure, like pressure in the body, that sort of thing.
Yes. Sorry, Karen. So it's not the pressure in the body. Definitely, they've got sensors which will feel that. But what's interesting about the Talin is where it sits inside the cell. So it's probably worth just introducing that ever so slightly. So all the cells in our body, almost all, 99.99%, are attached to this protein-like meshwork, which looks called the extracellular matrix. It's like a meshwork, and all our cells attach onto it, and that holds us in the right place. There's a bit of feedback. Is that on my end or yours?
It could be on my end. I guess I can put
anyway, so they're all attached, and inside the cell we've got this this protein Talin, and it helps the cells attach, but then it also connects this the adhesions. They're called adhesions, these things where our cells are attached. It couples them to the actin cytoskeleton, and the cytoskeleton is the key bit in this, because that's like actin and myosin. So in your muscles in your arm, the actin and myosins are arranged in a really ordered way to form muscles as we know it. But in every single one of our cells, and in every single cell going down to the slime molds and beyond, this actin myosin skeleton provides the cells with rigidity and the ability to generate forces and move things around within the cell. So Talin connected to this mechanical machine inside the cells. So the light bulb moment in terms of the MeshCode theory came from some experiments we were doing where we found that these Talins got here we go. So if you look at figure I don't know, 2 probably.
Are we at 2? We're at 1. At 2.
Yeah. So at the top of this figure, this is what this Talin molecule looks like. And the bit which we're really interested in are these R1, R2, R3. These are the switches. So these 13 switches are what we discovered can switch between 2 different states. And that's shown in this panel B. And it's really nice because you can hold one of these switches, you can pull on it, and it will switch to the 1 state. And then you let go of it and it will go back to the O state. So we can constantly switch these switches just by generating tiny little forces using the cell's contractile machinery. So you can have a signal which comes to the cell, causes a contraction and pulls and updates this switch pattern. And that was when we discovered these switches and then realized that they've got this capacity to serve as memory molecules, because the pattern of O's and 1s is stable as long as you're holding this molecule. So you can have a pattern of O11100 in the molecule, and it will last like that indefinitely as long as you can hold it still or stabilize the patterns with other molecules.
Okay, and you're showing 12 bits of information here, basically. Is that
12? Yes, that's right.
Okay, so so I'm wondering so outside of the synapse or outside of neurons, what do we know about how this information would be used in other types of cells? Is there any, how coarse grained is that? How fine grained is that? So what kind of information?
This is where the notion that this might be some sort of machine code underpinning how we work, because these proteins are essential for holding all of our cells into the correct places. And our initial work was on cancer cell migration and this process called metastasis, where cancer cells, when they're growing in a lump, can be quite sedentary, but then they become migratory and they suddenly start to crawl away. And there's specific triggers which drive that, and one of them is that the cancer gets stiffer, and that means when you're pulling on these switches, it's much easier for them to switch. So the cells start to run different programs. And our working hypothesis is that that's driven via these switches in Talin. And we can show that because if we deliberately soften these switches, make them so they unfold much easier, we can simulate that sort of behavior. So one of the things that we do a lot of is to try and reprogram this molecule to make the cells have different properties.
So that would be one of the magic bullets we're looking for cancer to keep it from metastasis? Possibly.
We'll hopefully find the targets to do that, because we can't manipulate the Talin molecule. Well, you can make drugs to do that, but we're doing it using mutations, introducing it ourselves. That would be hard to do. But if we can adjust the mechanical environment of the cells, or normalize it somehow to stop this mechanical coding, it's called mechanotransduction. If we could tone it down a bit, we might be able to treat cancers in a new way by just reprogramming the cells back towards a more normal sedentary state. So yes is the short answer, but it's slightly more complicated.
Okay, and still outside of the brain, if we're looking at morphogenesis,
yes,
embryogenesis, right? And we're looking at say we've got a epithelial sheet, and we're looking at how it breaks up and goes to mesenchymal cells and migrates, right?
Yeah.
But then there's also a possibility here of tissue type differentiation, in tissue lineages.
Yes.
And is it possible that this code is somehow involved in tissue differentiation?
Yes.
Type
that's really good. That's actually one of the best pieces of evidence that this is a code which is underpinning that, because there's a beautiful experiment from Dennis Disher's lab from a guy called Adam Engler about 15 years ago or something, where they take some stem cells which could in principle differentiate into any type of cell type, and they put them onto an extracellular matrix in a petri dish, and the only thing they vary is the stiffness of the matrix that they put them on. So if you put them on something really, really soft like jelly, then that stem cell will sense that and reprogram into a fat cell or a brain cell. If you put them onto something like glass, then the mechanical environment is very different. It's much stiffer, and that will cause them to differentiate into a bone cell. So a major driver of what the cells do and how they respond and which genes they read is dictated via the physical environment. And we believe it's these switches, but it's this whole mechanotransduction machinery inside the cell, which ultimately gives rise to that reprogramming and those different genes being switched on and off.
So how much do we know about getting from the signal at the cell surface with these these talons are at the cell surface primarily, right?
On the bottom of the cells where they're attaching onto the underlying extracellular matrix. Yes.
Yeah, I think we've got do we have a figure for that?
Yeah. So here, if you look on the right-hand side at the, you've got this bit which says ECM. That's the matrix. So all the cells are on this matrix. And then those little tiny blue and light blue V shapes, they're called integrins. They're like the hands of the cell. They will physically grab onto the extracellular matrix. And then talins that's just inside the cell, where it can couple those complexes to the cytoskeleton, which is shown in red. So this is the cell's musculature, the cell's contractile machinery in red and yellow.
So the ECM for viewers is the outside of the cell, extracellular matrix.
Yeah.
And then the integrins are transmembrane structures.
Yes, right. Yes, very good. Yes. I should have said that.
Yep.
And the development of ECM was that, like, the transition from being unicellular to multicellular, you needed to they started to make cell-cell connections, But then they also started to lay down this extracellular matrix, which they can then grab onto, which can send instructions into the cell as well.
Okay. And then we have the tail ends connecting, and that's where you would end up with the signal. And do we know much about how the signal would then become either epigenetic or genetic expression?
Yes. So if you go to, on this figure, the tail ends are all wired up there in this meshwork, and there's a meshwork of other proteins which have got these switches in. That's why I decided to call it a mesh code rather than a straight up tailing code, because there's a meshwork of switches which make this code. But once this cell gets stimulated, it switches on the motors inside the cell, the myosin motors, which pull on this scaffold. And the idea is that that signal comes in, pulls, and then it flicks these switches and changes the pattern. But if you scroll down to the next figure, I think it is.
Here.
Yeah. So if you look on this left-hand side, the reason that these switches convert into a readout is because the reason that the switches, and the reason we discovered they were switches, is because each of those domains binds to different signaling molecules, and whether it's in the O state or in the 1 state. So in this top panel, I've drawn it with a green, an orange, and a yellow signaling molecule bound to those switches in the O state. But when you flick a switch, you don't just change the shape of the molecule, you also change the signaling complex, because you kick off the orange guy and you bring in the blue guy.
Right.
And that changes the signaling at that site. But also where it's interesting is that some of these signals which are bound to Talin are things like transcription factors and splicing factors. So when you kick these off, they then go towards the nucleus, and then they can start to change gene expression. And not just those, there's also a whole set of mechanosensitive transcription factors, like you might have come across YAP and TAS, and they go in depending on how much tension there is in the cytoskeleton. So all of these factors move around the cell depending on the mechanical linkages, and those mechanical linkages are all attached onto the tailings. So it's like a massive mechanical computer is how I envisage it.
Yeah. Yeah, that's from the standpoint of morphogenesis, that is beautiful. I mean, that is a rich bit of mechanism to look into.
Yes.
Because there's a whole lot of mystery on these cell lineages and how they differentiate, and how they
work being done on that, and we're moving fast on the as a scientist, I would say, on how mechanical cues in conjunction with chemical cues and morphogens and signaling pathways all give rise to body plans and the formation of complex organisms.
Yes, yeah.
All distill, but it could, in the model I'm trying to present here, all distill down to a relatively simple set of rules in terms of the cytoskeleton and these switches changing the patterns and changing the cell's behavior. So each cell would have slightly different inputs and then reprogram to know what it's supposed to do. So talks of machine code and things like that become that's kind of where my answers in atheism versus creationism comes into play, because that kind of talk is used over and over again for intelligent design. So there's that. Yeah. So, but I mean, that's only if you want to see it that way. Like, I don't know. Right. I think both on the atheist extreme end and the creationist extreme end, I think there's so much which is agreed on. It does, like, you can have a machine code which is just because if it's a binary patterning across the whole cell, it's a machine code. Whether there's been a coder or whether it's just maybe the optimization, instead of being done by a coder or a programmer, is done by brute optimization, efficiency as the main driver. So I think that that's a bit of an artificial partisanship, really, if you ask me.
Yeah, it depends on where you want to take it.
Because if you want to say where I want to sit on that spectrum, I would say that I pivot between the two, because there's certainly bits which are missing from our like evolution from nothing is really hard for me to grasp, because well, from a cell I can imagine that you can build up complexity, but there's certain bits where you've got to capture something and then you've got to reproduce it, and certain bits where it might actually be unfavorable in the short term, but then longer term give rise to a favorable change. So it's hard.
Abiogenesis is, kind of the big bugaboo right now, as far as how did all of this get started, that sort of thing. But the interesting thing here is what you hit upon is the equivalent of the genetic code, the trinucleotide genetic code for another type of molecule, these talins, right? With a possibility of a complex code. But then, like, drawing back, I'm going to be talking to, are you familiar with Daniel Nicholson, Dr. Daniel Nicholson?
No.
He's a philosopher of science and biology, and also a molecular biologist. And he has written a paper called Is the Cell a Machine? And of course, he comes down on the idea that the cell is a machine. That it's not machine-like, that it's more stochastic and statistical in nature. And like, if we look at the reality of what we got here, how many talin molecules do we have in the average cell, right?
Yeah.
Do we have a scope for that? I mean,
there's like probably 100 in each adhesion. There's probably, I don't know, a few hundred adhesions in each cell. There's a lot. It's and like a machine, all of the computation we do, it's all been driven by efficiency and the need to become more efficient. Right. And maybe we just converge on the same solution because that is the most efficient way to do something.
Yeah.
Whether but I mean, it would be created or happen to come by a random choice. I think both are total faith-based.
But if there were, basically, if there were a statistical, like a statistical tendency for a lot of these talin molecules to be in one state, and then of course you'd have fringe states, right? But overall, for signaling in the cell, what's going to affect genetic expression is going to depend on the statistics of how many of these signal molecules we have, right? So I mean, it isn't quite real clean machine-like, but it's more statistical because not every one of these talin molecules is going to have exactly the same code.
Well, I don't know, because my view of this, and I'm trying to publish a kind of follow-up, but like, is that there's probably within a cell, there's probably solutions to all of these switches and all of this cytoskeleton and all of these different motor proteins where it balances out, and those balanced states are probably certain patterns, which is what we really like to try and prove. So, and that's where I think each individual talin molecule is part of a big system. So at the end of the calculation, the patterns will be fixed across all of the talins to give a proper solution. But it's not explicitly dependent on each of these switches, because on any one switch being in, because the problem is I saw a really nice talk a few years ago where they were talking about how you make a lung. It's a bit of a detour, this, but it was this branching morphogenesis where a capillary goes, then it divides, and then it divides, and it divides. Right. And if you try and do that, the obvious one is it's like a fractal, because it keeps doubling and doubling and doubling. But you can't, if you try and run that as a simulation, as soon as it hits a little bit of gristle or hits a bit of noise, it just piles up and it becomes a horrible mess. So you can't rely on perfection. What you need to rely on is a solution which can tolerate imperfection. Right. I think that's what the body does. The morphogenesis and the body plan and the brain all do so well, is that it's got this statistical component, but also that there's it can readjust if there's an issue, or to give a solution, is what I'm trying to say, probably. Yeah.
And that's kind of where the intersection of biosemiotics and information and biology is becoming a really interesting new way of looking at things. Yeah. We're looking at we're looking at a system. I was listening to Terrence Deacon. I don't know if you're familiar with him.
No.
An incredible person who has a very, very radical idea about biology that is making a whole lot of sense in this last decade. He wrote a book called Incomplete Nature, and he talks about how biological systems are robust because they are not machine-like, because they are not, because they are more statistical and stochastic in nature, and there are solutions in the space always, that can be found that would not be found if it was exactly precise. So he pushes against precision,
Yes.
that sort of thing.
So I switch again. I flip between these two because now I'm at the state where I can imagine, based on these switches being part of these big networks of things, that I could imagine that as each cell jostles and tries to fit, that there's a unique solution to it where every single molecule has got a specific shape, and within that you can have variation, because if one comes loose or goes into it, it'll readjust. But you've basically got these little tiny interdependencies, which that's like in a city really, where the day-to-day bits of all the parts aren't necessarily tightly regulated, but across the whole of the city it works pretty nicely. And that would be what I would imagine across all of our cells in our body, where they're all working to the same plan, but it can really tolerate small unexpected changes,
perturbations, basically. Perturbations are just absorbed, right? Yeah, which is really, really interesting. I've been looking into the evolution of the liver, and the first thing I ran into is this tissue type, hepatocytes, on each corner have sort of an actin structure on the corners that shape the cell into what it is. And my question was, how in the hell does that happen? and basically, so when you go from one corner to the middle of the cell, you have a variation, and this code that you're talking about is a possibility for that variation. Looking at the variation from along a cell membrane, how cell membranes are segmented, right? And divided up so that the cell has a specialization on the surface, that sort of thing. And you have different, different receptors and all of that. And this is really exciting to me, because this looks like the possibility of just unbelievable variation, right? Because of this code, that sort of thing. So anyway,
the talins are just the one bit at the bottom of the cell, but it's connected to on the nuclear envelope. There's other things which have got similar switches, not as many, like the Nespins or the link complexes and all of these type of other switches. So you could easily imagine with some switches at the bottom and at the top, that as you push and pull on them, you can be updating across the whole length, across a big scale across a cell. From the front of the cell, all these triangular actin structures, to under the cell where you've got these talins where it's getting the nucleus on top. So you can easily imagine this incredible versatility in this system with these rules of how actin polymerizes and how these switches and proteins interact with it. So I was just reading a paper on contact points between organelles, like the Golgi apparatus, mitochondria, endoplasmic reticulum, and all of these contact points. And there again, actin and microtubules are involved in actually supporting these structures inside the cell. Because a question is, how does the Golgi apparatus assemble itself and stay that way? And then along this line, you have the possibility of this entirely signaling architecture, coming from the surface, which is pretty cool. I think the whole, my view is, like, a lot of what we know has been mapped out from a cell in culture. And so we learned so much about how it works, but the big missing piece of the cell in culture is it never makes all of the correct contacts with its neighbors. It never gets to remake these the matrix into the pattern it wants. So it never reaches a final state. So it always looks chaotic and dynamic, and the organelles are moving around. But I think in an actual tissue, these things are much more positioned, almost to the point, I think, where the cytoskeleton is ordered and it actually holds all of these organelles where it needs to be. Like in a synapse, you hold the mitochondria right there, so they can be generating energy on tap right at that site. So everything is, so I think it's not it's not random or as chaotic as what people say, which is why I'm moving to a position where I probably disagree more with that Terence Deakin's position, too, that this is a mechanical computer. This is a machine. Just it's not made of metal and silicon. It's made of protein and fat.
Well, I think we'll all get along if we stick with stochastic statistical codes rather than precise, perfectly designed codes, that would be rigid, and as long as it is statistical. But before we move on from that point, I want to mention one of the things that when you have a cell lineage, that is basically developing and moving along, and it has endothelial to mesenchymal, blows apart, and then these tissues end up in a like in the liver, it will end up in proximity of the heart, and the heart has an effect on it becoming a liver. Yeah. If there's no heart, there's no liver developed, right? That sort of thing. But you also have cell history, and you may have hit upon the thing that gives the differentiating cell a bit of history about where it came from, about where it was before, right?
Yeah.
So you have a historical aspect to development, which would tell us why it doesn't work in the petri dish.
Yes, and the picture, it never comes into contact. It never reaches its end state, like its energy minimized state, because it's constantly moving. And the reason it looks so dynamic in that state is because it's constantly trying to feel and trying to work out what it's supposed to do.
Right.
And yeah, I'd say that these switches provide a way to encode memory on a relatively dynamic scale. But then because they're directly interacting via mechanotransduction, changing the genes, changing even the epigenetic marking of the genes. So you can encode something quite quick in a switch, which then leads to more long-lasting changes by reprogramming the actual which genes are available and which ones are tightly wrapped up in the nucleus. So yeah, I think there's different layers of memory in a cell, going right back, probably all the way back to the first cell, as it develops, like they're constantly updating and changing how they are. So yeah.
I've got I'm not sure what you want to do with YouTube and YouTube presence, but it's kind of become a thing for a lot of scientists to get on YouTube and start to do some hangouts. But it would be fun to put you in the same room with Daniel Nicholson. The cell is not a machine, and kind of work some of that out, because there is something there. There is a solution to those two camps. That is something new in between that I'm very, very interested in.
Yes.
And somehow the biosemiotics people are trying to nudge their way in and make sense about it all.
I was going to say that a machine is, I'm not trying to say it's like a train or a steam train, or it's not. It's incredibly dynamic. It constantly disassembles and reassembles. So it's nothing like our mental image of a machine, but it happens with really high reproducibility. The same thing happens every single time, because look at us, we fertilize that egg, and then 9 months later, here we are, 99% or 99.99% of the time. Every single thing unpacks in a beautifully ordered, logical way to give rise to us.
And that's where you would agree with Terence Deacon, and that he says, no matter how you perturb this system, it settles back into that groove. It goes in that groove over and over again. And so there's something pulling it back into the shape with itself. And he sees it as a dynamical system. Very complicated.
All of the bits of it is just interdependencies, like little feedback loops. So at the adhesion, you've got these feedback loops, but then they're connected to other things further away. So the whole system is constantly self-correcting across different levels, from the individual proteins to the adhesions, to the linkages, to the cells, to the neighboring cells, across the whole tissue. The whole thing is with different levels of interdependence and interdependability, or whatever the word is. Like, they're not, if you change something over at the top of your arm, your fingers aren't necessarily going to have a massive reaction, but overall the whole system is constantly, it's all interdependent.
Yeah, and he looks at it as over many small overlapping dynamical systems, basically, that all end up together in these, like if you were to try to draw circles around it, you would have circles around the whole thing and circles in the middle and tiny circles and, you know, just all these overlapping patterns. And it ends up being this self-correcting system, which he calls life, which is how he defines the difference between a living system and, say, some other system that's going to be differently.
No, and I think, yeah, maybe I don't disagree with any of these people, and even the argument on the machine or not machine. Yeah, I'm not a big it's a machine. It's just it's got machine-like characteristics because of this very quantized, and the good thing about switches is that you can have a lot of stuff going on and you can have a lot of small changes, but at the end of the day, a switch is either in that state or it's in that state. Right. So it buffers against it. But because, and a good example is on Talin, there's two switches next door to each other, one which upregulates contractility and one which downregulates. So even if you kick off too much of one, it will turn up the contractility or down. So it can constantly be correcting. So the whole thing, it works out. So in some respects, going into this binary coding notion oversimplifies it because it's the signals which interact with the code, which is where the magic happens and where the variability occurs. But at the end of the day, it is all those switches which are coordinating that, in my view.
Yeah, I think a good term would be over-reduces. Like I'm always looking for, you know, I'm looking at one particular or two particular proteins interacting, and I'm coming up with this reductive pathway. And sometimes when you focus on this one pathway, you're over-reducing, and you have to look to this wider thing. And there's this tension between the two because of course we know that proteins, you know, fit together and do this thing. So we know that we can reduce the mechanism to this pathway. Yet we're being told that, you know, this it isn't quite that pat. You know, it's more stochastic and there's more bumps in the night than we're accepting when we over reduce something. So there's a tension between the two ideas.
And I'd say here, like in terms of these switches, then a single bit of a molecule will be in one shape, and then if you exert force on it, it will change to a different shape. The proteins which interact with those two states, it changes the affinity, how strong they bind, by 10,000 times, whether it's in the one state or the other state. So ligands which bind to the O state bind, as soon as you unfold it, there's no binding site for those anymore. And the same with the ones which bind to the unfolded state. When it's folded, that binding site's not available. It's inside the domain. So you can change, you can have a protein-protein interaction, which is changing really with the same two proteins in close proximity, depending if the cell receives a signal which makes the contraction which pulls to flick that switch. So it's really complicated, and this sort of notion of stochastic and these protein-protein interactions, yes, that's very over reductionist, but that's the trick. You take it down to a very reduced state. Like we do a lot of work in a test tube, protein-protein interactions. And then you can build it back up because you can take a protein-protein interaction, understand the interface, put a mutant in it so it no longer does that, and then look in a cell or look in even in an organoid or even in an animal and look what happens to that cell if that interaction is no longer possible. So you take it down to the bare bones. Like we only go as far as the individual proteins. You don't need to go down to the individual atoms or anything. But from building it down, reductionist, and then build it back together. So I think if you were to just stay on one level of explanation, I think you would be in trouble and you would get down dark alleys. But if you keep building it up and then looking where it fits across these different levels of explanation, then I think any single thing, whether you're looking solely in an animal or solely biochemically, as long as you're collaborating and transitioning between them, I think you can find useful stuff out.
Yeah, I look at, I guess when I dabble in the philosophy of physicalism, there's a reductive physicalism, and I contrast it with what I call constructive physicalism. And basically, we're always looking, you know, we understand by reducing, but to truly understand what is happening, you have to construct, and you have to move back in the other direction and bring these systems back into their totality and stuff.
And what's coming on really good at the minute is there's loads and loads of, like, and it's it's cheap as chips to sequence people now. So everyone who presents with a disease or a condition, sequence them, find what's gone, what the defective bit, and then you can do the biochemistry and you can say, oh, it's because of this Talin no longer interacts correctly with kindling or whatever the mutation is. So you can work it out, and then so you can learn a lot from that because you understand the nuts and bolts, and then you get a sequence which tells you one of the nuts is broken or not working properly.
Yeah.
So it's across all the layers, definitely.
Did you use the phrase cheap as chips?
Well, is that not a good phrase?
No, I love it.
It's like I love it. Correct same sequence a human is like under £1,000 now. Like when they did the Human Genome Project, I don't know how many billions that cost, but now it's down to £1,000 and you can get a sequence of a person. So hospitals can just do that when you present. They even sequence your cancer.
No, I love it. I wrote it down. Cheap as chips. I like it.
Maybe that's an English term. I don't know.
Anyway, fish and chips.
You're steering very clear of the brain part.
Yeah. Okay. Now we're getting down to the brain part. So the problem I had, I look at the brain and mind as being a dynamical interaction with whatever's in the world right now, in our body, and all around us, basically. And I see that the brain and mind is pretty much driven by the external inputs to the brain, and that's sort of what drives it, right? And when I look at something like Penrose talking about quantum effects in microtubules, I'm very skeptical because it hashes out to, you've got an axon, and you've got a neuron, and you've got 10,000 inputs or 1,000 inputs or whatever being summed into across the axon, the hillock, and where it actually fires the axon, and you end up with one bit of information on the other side to some degree, but it's not exactly a bit. It's more of an analog signal, because you're going to have a certain amount of strength, right? But then you're going to the next neuron, and of course you have 10,000 other inputs on that one, on its dendrites. And when you talk about now, when the common man hears the word, that there's some sort of information in the synapse, they automatically think in terms of representation in the mind. Like somehow the mind has all of this data, all of this information that represents how my cup looks. This cup I call my red kitty qualia cup, or qualia. When I talk about consciousness, because I use it as an example of how we visualize stuff. But when I have this cup in front of you and you can see it visually, okay, it's one thing. But when I take it away and you're just imagining the cup, it's a very, very different world. So anyway, I tend to push back on this idea of eidetic representation in the mind. And then you come along and you try to topple it, and so what do we have in terms of, okay, but then there's this problem of what is it, post-tetanic potentiation, where when you run a spike train on a neuron, okay, it's very, very likely to remember that for some period of time. And if you run enough spike trains on the neuron, or on where the axon itself, the post-synaptic dendrite has, I mean, we can understand how that can remodel, but the presynaptic axon, basically, we weren't clear about how that could remodel. And what you have here is the possibility of seeing how it can remodel and how it can remember a spike train, or get temporal information along with the fact that the neuron just fired. You see what I mean? Mm-hmm. So anyway, I would caution you when you talk to pop writers, about your theory, that you don't get pigeonholed into some sort of a weird representation of information in the brain in these codes. Yes. And that we look more in terms of the dynamical system, what it is to be a mind in the world.
Yeah, the representation stuff and all of that, I tend to stay wide of, because how I'm viewing this is that this is just classical cell signaling, where you have one cell talking to another cell, it sends a signal and it causes the contractility in the next cell, which just alters the pattern. So one cell, it's just basically tapping it and telling the next cell to change. Because when you see something, you've got, I don't know how many optical nerves, lots of rods and cones feeding the signal in.
Right.
It's all just a binary set of information, but it just updates these patterns because that cell is wired up to a circuit of other cells. So it will send a signal into them. Yeah. And then that one will react, and the whole thing will just constantly update and tune itself. The shapes of these molecules will change. But the big thing, and what we're really keen to prove, is that these in a cancer cell, it's really clear that if we change the pattern of the Talin, we change the signaling of that cell. And we can stimulate the cell with signals and make it change its patterns. We can see all of that. That's fine. It's the trick is to see that in a synapse, and that's what we're currently doing at the minute. We want to try and prove that you get these shape changes across a synapse, which then changes the activity of that synapse, either up or down. Yeah. And also the ones in close proximity and further afield, like what the actual rules are. Because we've got these memory molecules, we know we can show experimentally that we can write information into them, because we can take a single molecule and write information into it. And we know that they're in the synapses. We know that they coordinate signaling. So the big bit missing is to show that they coordinate signaling as a function of their shape in a synapse. And that's what we're currently trying to do. Because once you know that, then you can constantly just build that this cell signaling, these cells all talking to each other, just updates them all. So you're just leveraging this mechanical computation across all of these different things and all of the different inputs. When I see your nice pink cup and red cup, also I've got all of my own associations or whatever with a red cup. So I start to, all of that bears down on it at the same point. But that's because it triggers these circuits. I mean, I think it's just a relatively simple set of just rules, and it doesn't require. I'm very skeptical about the quantum stuff, if I'm honest, because it's complex and it doesn't explain a simple case. Whereas here, you could imagine, and even in the case of a simple organism, you could be constantly updating. You could see this quite well, I think, and that's another thing which we're trying to do.
Okay, so when a neuron fires, when the axon basically fires onto across the synapse, there's an actual mechanical change in the cell membrane as a result of that firing?
Yes, so it's pretty well established. The cool thing about all of this is, if you look back through the literature, this has been, I've just joined up a lot of dots, but there's when you stimulate a synapse, you get contractility of the cytoskeleton on the pre-side, and the contractility on the presynaptic side is required to cause the vesicular release. So that's what in this panel B, I try and suggest that you might be able to pull and release a switch and actually release a vesicle or trigger the vesicle release. But on the postsynaptic side, also, it's well mapped out that the cytoskeleton changes when you stimulate a synapse. And the cytoskeleton changes as you stimulate a Talin in a normal cell. So we can start to build. So that's all well known. What's also really well known is that the ECM is essential for all of neuronal processes, and integrins are essential for that. So all of the long-term potentiation, all of the plasticity, and all of the stuff which everyone knows and loves are all explicitly dependent on integrins and the ECM, and they all drive changes in the cytoskeleton. So the bit here is that the bit in the middle, which is coordinating that, is this molecule which has got these switches which are able to capture that information in a really fast way and in a quantized way as well.
Yeah, like my neuroscience is from 15 years ago, and it was sort of a big, it was a big thing about how the presynaptic neuron could rewire, and you have a solution here for both pre and postsynaptic reorganization, right? Depending on the reason I mentioned this book to you, Spikes, is because this is about the spike code in the brain, and it's done by a biophysicist and a couple of neuroscientists, and exploring the neural code basically. But when neurons rewire when they get certain spike trains, and these spike trains are extremely noisy. If you look at them, they don't make any sense until you start statistically looking at them, and then you start to find a thing like when you look at this cup. Okay, it's in one part of your visual field. Now it's in another part of your visual field, but things stay constant. So there's a whole bunch of statistical noise reduction going on in your visual system.
But also in that, there's also quite a lot of what's becoming apparent is that a lot of that information is first encoded in the eye itself. So in the sensory part feeding into the nerves, and in the synapses connecting there's information and then that's talking to your brain. So in this model, you're constantly updating all of these synaptic points, all of these junctions where things come together with that latest information. And now, because I'm sort of even a bit primed that you might get your cup out again, I can start to think like I'm already sort of anticipating it almost. So I was ready, you see. So and it would be constantly being updated at all of the junctions, and even down to your muscular junctions, neuromuscular junctions. So that's
there's a lot. There's a lot being. I think his name. He got his Jeffrey Hawkins. He does this stuff on AI in the brain, and he talks a great deal about how the cortex is basically a prediction machine. That it always wires across, like particularly at layer 1. There's all this cross wiring. That basically when you see,
because that would be useful here, because the map of the cortex is beautiful.
What was that?
The figure 5 of this paper.
Oh, okay.
It's easy to scroll down to.
Figure 4. We'll go to figure 5. Okay, right here. Yep.
So maybe that one, but even more so is figure 6 then, the one of actually what the cortex looks like, because it's directly relevant to what you're about to say, I think.
Let's see if I can get a little more of that figure visible. Okay, that's figure 5.
Yeah, so that one's just the idea that once you do show me the red cup, my eyes detect that, but even in the eye, in the nerves and the synapses in my eye, they are still picking up and changing as updating, and then feeding into the circuitry of the brain, and the whole thing is constantly just updating this register. But go to figure 6. Sorry, that's what I meant. I haven't read this paper for a while.
Okay, we're figure 6. Oh, right here. Yes, the brain.
Yes. So this is the cortex, which you're saying about the structure of it, and it's incredibly well ordered.
Yep. Yeah, and with these pyramidal cells, this layer 1 up here, I don't think you can see my cursor, but the very first layer at the top, there's all this cross connection going on between cortical columns, and then at the bottom, of course, we've got the thalamus, or the thalamal cortical, or what is it, thalamic circuit, right? It basically, there's this feedback with the thalamus, but there's some incredible things that happen with the thalamus. When you first see the cup, you see your thalamus first recognizes red blotch, and then it starts to work down and starts to through lateral inhibition, it starts to narrow down the focus to, oh, that's a cup. Oh, that's a coffee cup, and oh, that's the coffee cup that Speed was holding up 3 minutes ago. So there's this whole, little perturbed circuit that hones in on what you're seeing very, very quickly. And of course, it makes predictions. And then, of course, you've got, the memory of just having seen it. Now, when you go to sleep tonight, you might even integrate my red kitty qualia cup into your long-term memory, and you will be plagued for the rest of your life with the memory of this cup.
I always remember it. That's right. And that's why I think there has to be an addressable read-write memory. You've got to be storing it in this very ordered fashion to be able to call it that quickly. If it was just a big network, a neural network, it's all just whizzing around, it's very hard to imagine where that information resides. Whereas here, it would be physically written, and there would be some sort of allocation, so that hippocampus would know where in that it was. So when we stimulate it, we immediately call the right memories, which then link to the inputs. And then over time, like you say, at night, then the body, your brain then makes that consistent with what you've got in the day, with what's already written, and updates it where required, and loses the rest of the stuff you forget. Is really important, because otherwise you would just get totally filled up.
Yeah, the hippocampus, I think, is a place where you're going to find the very quick read-write mechanisms, you know, that are going to, I've always had a problem remembering Terence Deacon's name for some reason. I can never remember his last name because I keep thinking it's some sort of a French last name, and it's not.
Yes.
And so
that with some other piece of information, and it steers you off. That. Yeah.
So I wrote it down last night on this on this pad I have here, and then of course I went over and started watching videos, and then I started watching something else or checking Netflix, and then I'm like, okay, Terence, I forgot his last name again. So I walked over here to my computer, and as I was approaching this newsprint pad, all I did was glimpse the newsprint pad, and I remembered Deacon.
Yeah. No, it's amazing.
and that's the hippocampus, right?
I think it's it's just amazing how your brain works, how it brings in all this information. And I think there's a tendency for a lot of people to oversimplify it, like that, the amount of associations that, or the amount of information it's using at the same time to make that real in our world. But the cool thing about the MeshCode theory and the hypothesis is if it's updating each of the synapses in those cortical columns, not only is it updating information, it's also updating the flow of traffic through them. So a bit like in the SD card on the left, when you call the information back, the flow of electricity comes as a function of the patterns of information. So I was actually stuck in Germany when I made this association between what we've designed with SD cards and the Cortex. It was at the start of lockdown in March 2020, and I got locked in my room for 24 hours. So I was just doing some internet research into how these switches could be used. And I think it's incredible how similar our architecture is, where you have lots and lots of repeating columns with very repetitive architecture, but each one's addressable slightly differently. So I think, that's my view of the brain. And that would also imply that information is written into specific parts of the brain in a way that it can be rapidly recalled. Not that the picture of the red cup's written in one specific synapse, but the change of the information is part of this mathematical representation, which is written into there, such that when it's called, the information comes through the circuitry and then appears in where I can use it. So
and then there's this thing about you got like a mini cortical column. Joaquin Fuster is probably, he knows more about the cortex, I think, than any other man alive. He's a neuroscientist. And we have all these mini cortical columns, and I think we got I can't remember, about 30 million of them or something like that. I can't remember what the number. And anyway, they are used in more than one memory, depending on which cross associations are hit, right? And so you have the same thing, and every now and then it will leak, and you will think about one thing, and then all of a sudden you'll remember something, tangential, way up, just way off the wall.
That might also be by design as well, because it's the cross links and the crosses, the bringing together piece of information, which is why we can think laterally and so creatively, because we're not just locked on one solution. We bring in information, and a lot of it might turn out to be rubbish, but then you might make a linkage which might be useful.
Why we can think intuitively.
Yes, and it might be that all of our inputs, all of our senses and all of our outputs, all of our muscles and stuff is just linked via this calculation and mathematical representation, is what I try and describe in the paper, where your brain wires up in utero into like a default state. But then when you are born, well, even when you're still in the womb, actually, but when you're born, all of the different inputs, whether you're in the desert or here in Kent or whatever, and what foods you get and what you're taught all wires up, and so you end up connecting all of your inputs and your outputs via this computation, and it's a mathematical representation is what we're talking about, written into these patterns of information, written in the shapes of molecules.
As a 70-year-old, I find there's a lot of, particularly a 70-year-old trying to understand molecular cell biology. There are all these memory problems that you run into as you get older. And when young people accuse me of, oh, you're getting old and your memory's starting to go, I tell them, no, the problem I have is that my brain has a lot of information in it. It's full. Yours has nothing in it yet.
Yeah.
Basically, when you fill it up, you'll be able to start, actually understanding what I'm talking about. But so you got locked in your room for 24 hours and you came up with this.
I was supposed to go into a brain conference in Leibniz, and all of a sudden it was getting quite close to, and then the UK said come home, because it was just as it went really took off. So
red.
I stayed in my room until I could get the next flight home. So I missed the conference, but spent the whole session writing this. So it was very good. I mean, yeah.
I think locking scientists in a room for 24 hours is a good idea. You come up with a lot of new research that way.
Yeah, definitely. Just that time without the distractions. Anyway.
Yeah. Yep. Okay. So, I don't know. I don't think we disagree too much. I would just caution you on this, on how pop science is going to see information in the neuron. And the information that's in the neuron is not the kind of information that we would imagine it to be, like there's, some binary representation of this cup. That's not exactly what's happening. It's an entirely more dynamical system than that.
Yeah, it's dynamic. The whole thing's dynamic. And that's why as soon I talk about it in there, like hypoxia, or when you die and these motors stop being dynamic and they start updating or holding this information, that very quickly you start to, you can't regenerate, even though your brain is visually perfectly okay. The information is gone because it's lost its synchronization. So that's why you can't revive someone who's had hypoxia for too long, because the brain isn't functioning and it's not connecting the inputs and outputs correctly.
Yep.
So and I think it does lead to a cool model for what's going on in the sleep, that electrical activity is physically just updating these patterns, generating contractility and pushing and pulling on these switches to update them to a meaningful updated pattern. So this is amazing. Like I say, when I first heard it, I was like skeptical, and then I started reading your paper, and it's like, oh hell, this can solve a lot of problems. I mean, there's a lot to be learned here, and really looking forward to whatever you do next. So yes, hopefully we can prove it. And if we can find one of these, the main thing we want to find at the minute is neuronal regulators, which are known to be important, which are interacting directly with these switches to try and correlate the wide body of existing knowledge with mechanical computation and mechanical memory. So that's what we're doing at the minute. That's how
you're gonna have to get some rats and mice and do a little in vivo observation. Hippocampus is where you're going to find your neurons that are going to be the most malleable.
Yeah. Although I think so far we've not done any. It's not involved. We've used half of a mouse's brain, one hemisphere, to try and find the new ligands, which is an experiment which we're doing at the minute. But most of it we can work out looking, because there's a lot to understand. But once we get these rules, we know the rules of a single memory molecule. So now we're looking at synapses in living synapses in culture to show that we can stimulate them and actually update these patterns long before we start to cut. Because I'm not a massive fan of animal studies for the sake of it, which a lot of this seems to be, where they're looking for what they're hoping to find. They don't know what it's. So yeah, at some point we'll need to go into that, but I think we can work out most of the rules before we go in.
Yeah, we want to thank that one mouse for his service, by the way.
And it was an offcut from a neuroscience lab who were using the other half for something else. So
he'd been in survey. There you go. He's a veteran.
Yeah.
Okay, so we're coming up a little past 1 hour, so we can end this here, and hopefully we can talk again soon. But
sure, this has been interesting.
But hang around, don't go away. I'm just going to end the recording. So, and I always say goodbye, cruel world, when I end my recording. So goodbye, cruel world.